So what I want to do now is I want to shift gears and talk a little bit more about populations and population structure. Typically, when we think about populations in the context of academic research, we're thinking about biparental populations. So in tomato, that would be F2s or RILs or back crosses. In potato, you're doing most of your mapping in F1s. Okay, so two heterozygous parents are crossed together, and then you've got your mapping population. But in the context of a breeding program, most programs have population structures that consist of many independent crosses. Okay, and I think that the lesson that we can learn from association mapping is that we do in fact have tools where we can start thinking about linking markers to traits, not in the context of a single biparental population, but in a more complex population. Okay, and I'm just going to run through some of the standard populations and then some of the populations that are now being used for what can loosely be called association mapping. So again, for inbred crops, F2s are standards. For outbreeding, crossing two parents together and looking at the segregation in the first generation. RILs and backcross progeny, advanced backcross and inbred backcross progeny have all been used. I think we could call those standard at this point. Emerging are natural populations, unstructured populations, family-based populations, chain crosses, and then nested association mapping populations, which are essentially nested RIL type populations, right? And I think some of these structures or ideas could easily be adapted to potato breeding populations. Okay. So some of the populations that have been standard have the advantage of having a strong theoretical basis. In other words, there is off-the-shelf software that can handle the population structures inherited in them. They have balanced designs, so you don't have skewed numbers in the different marker classes. There are tools for interval mapping, so really what interval mapping brings to the table is it changes the point of analysis from a single marker to a pair of markers and then incorporates recombination distance. The problem from a breeding perspective is that most breeding programs don't collect extensive data in those early generation populations. So in a tomato breeding population, it would be extremely rare to collect a lot of data on individual plants in that first generation. And then the other thing is that these standard populations often retain way too much donor parent. Okay? And this is particularly a problem with integration. And I know that there's at least a couple of people at this meeting that are doing integration work to bring in new resistance into potato. And so in inbred species where we're doing seed transmission, it's very common now to use advanced backcross or inbred backcross type populations. And these have some advantages in terms of reducing donor parent and creating a nice uniform genetic background, but then they have disadvantages in terms of an unbalanced design relative to the marker classes, and therefore also very limited power to detect epistasis. And so those limitations have really fueled the interest in the unstructured populations, because these are really populations that are more like what we would use as breeders. They may be unbalanced and therefore have some of the disadvantages of an unbalanced population, and we need to correct for structure, and I'll explain that in a little bit. Okay, so what I'm trying to do here is just give you an overview of some of the pros and cons of some different population approaches, just to get you thinking about that.